So today I will try and talk quite slowly, so hopefully you can understand my English. I will talk to you about NULISA. And NULISA is a new ELISA, or it is also nucleic acid-linked immunosandwich assay. And this is a new breakthrough proteomic technology for fully automated, ultra-sensitive proteomic biomarker platform for profiling biofluids like blood, CSF, or cell culture lysates. Alamar was founded 7 years ago by Yuling Liu, a Chinese entrepreneur, and his team of scientists who have previously founded 3 companies, including a company called Panomics that was bought by Affymetrix, and then a company called Advanced Cell Diagnostics, which was bought by Bio-Techne. When Yuling founded Alamar, he had one simple mission, and that was to enable precision proteomics to power the earliest detection of disease. To do this, Yuling and his team of scientists understand that there were some gaps in the current technologies available for measuring proteins. And so they set about developing a new technology that could change the game in terms of sensitivity, multiplexing, and automation. And so to achieve the mission of enabling early detection of disease, the Alamar team has invented NULISA, which is an attomolar sensitivity, so a femtogram per mil levels of detection of proteins using a next generation sequencing readout. And it enables highly sensitive protein analysis at a very large range of multiplex levels, whether it's a single protein or whether it's hundreds of proteins at the same time. I will describe in this presentation how NULISA works and how we achieve such high sensitivity with the Alamar platform. Also at Alamar, we have invented a technology, a trademark technology called AttoBody. And these are nanobodies or biparatopic antibodies, which are capable of exceptionally high specificity. And the combination of these antibodies and the NULISA chemistry enables unprecedented levels of sensitivity and specificity for measuring proteins. Finally, and very importantly, Alamar set about developing a fully automated instrument called the Argo HT, or high throughput. And this instrument was designed with diagnostics in mind. The idea being that we wanted to create a proteomic workflow for the first time where you could load samples at one end and then come back with a fully automated workflow to the data without any manual steps and with exceptionally high reproducibility. So during this presentation, I will talk to you about the Argo HT instrument and how that works. So Alamar is focused on automated solutions for ultra-sensitive protein analysis across a range of multiplex levels, so we can detect critical clinical biomarkers and take them all the way through from discovery in large cohorts to the clinic. We have 175 employees now and are generating very large amounts of revenue since our launch at the beginning of 2024. When we launched the technology, we published this paper in Nature Communications called NULISA, a proteomic liquid biopsy technology with attomolar sensitivity and high multiplexing. And in this publication, you can read in good detail how the Alamar technology works. The 4 main advantages of the Alamar technology are listed here on this slide. So first and foremost, this is the most sensitive proteomic technology available. In customer testing, we have seen that it is 10 times more sensitive than Quanterix, and in many cases, 1,000 times more sensitive than Olink. And this enables you to quantify very low abundant proteins with exceptional levels of precision. The second feature is that NULISA has the widest dynamic range of any proteomic platform. And as you know, if you try and measure proteins in a blood sample, that is very challenging because you have proteins like albumin or fibrinogen that are very, very highly abundant and take up 95% of the protein mass in a plasma sample, for example. And so you need to have a very broad dynamic range and a very high sensitivity so you can measure low abundant and high abundant proteins in the same experiment without any sample manipulation or dilution. The third important part of NULISA is that it is very flexible. So you can either look at a single protein, let's say you want to measure phosphorylated tau 217, you can do that in a single assay with exceptionally high sensitivity. But you can also measure hundreds of proteins from a single experiment from a very small amount of sample, 10 microliters. And you can look at hundreds of proteins at the same time. And you can look at any combination within that range. And so this means you can now run large panels and smaller panels or single proteins in one easy experiment with one instrument. And the final thing is the full levels of automation and the ease of use of the Argo instrument. So I will show you the Argo instrument, but everything is designed to make it minimal hands-on time and exceptionally high reproducibility. In this paper, Nature Communications, the platform is compared to Quanterix, Olink, and Mesoscale Discovery, and you can see the improvements that the NULISA chemistry enables. Today, I will talk a lot about sensitivity, and sensitivity is really the key to being able to diagnose diseases early. And what you can see here on the left is a graph showing that with NULISA versus a traditional ELISA technology, you can achieve 10,000-fold improvement in the limit of detection. And so the NULISA chemistry is one that really involves removal of background to enable the lowest limits of detection to be measured. And this is really important because it allows you to detect critical disease biomarkers earlier in disease and at lower levels. And it also enables you to measure key proteins in healthy individuals as well as disease individuals. So if you're looking for changes in protein expression when someone's developed a disease or is moving through stages of a drug treatment, then you can measure these at all stages of that process. And using this technology, we've been able to discover novel biomarkers and drug targets that are currently simply not measurable by other current technologies. So now I'm going to talk about the Argo HT. We call this proteomics at the push of a button. And what you can see here is our benchtop instrument in the middle, the Argo HT. And this instrument sits on a bench and has 3 bays that you can load 96 well plates of samples into each of these bays and then walk away and come back to the data 24 hours later. So the throughput of this instrument is almost 300 samples per day with no manual intervention. On board this instrument, there is a qPCR machine that will do the readout for single-plex or low-plex assays. And also on the Argo instrument, you do, for the large multiplex assays, you do every step of the process automatically, including the library preparation for next generation sequencing, which is the readout for the large panels. All of the reagents and all of the plates and tips and the labware required to run a 96-well plate is provided by Alamar and QC'd by Alamar. And everything is also lyophilized or dried down so that we can ship and store all these reagents at room temperature. We also provide software so you can basically go from your sample into the machine, and then the machine generates the data for you to do the analysis in the cloud, where you can do statistical analysis and understand your data very quickly. So this is truly a revolution in proteomic workflow, where all you need to do is load your plate of samples, and this takes less than 30 minutes of hands-on time, and we provide the sample plate for you to put your samples into. And these samples can be plasma or serum or CSF or tissue homogenates, and they get loaded into this 96-well plate format. You then load your sample plate and all the reagents that come in the kit onto the Argo instrument and press go. If you're running a singleplex assay, for example, the results will be generated from that 96-well plate in less than 8 hours using the qPCR machine inside the Argo. If you're running one of our broad panels, for example, the inflammation panel, which I'll show you in a minute, then the Argo will do every step up to the point of library preparation and pooling everything into a single library. You then take that from the plate and load it onto a sequencer. And this can be an Illumina sequencer or an Element sequencer. And then you perform the analysis with our cloud-based NuLISA analysis software. So every step of this process is very simple. It doesn't require very extensive training. And I believe this is going to be available very soon also at Phila Korea. So you'll be able to generate data through our partner lab in South Korea. So what we are focused on at Alamar is being able to take you through the translational journey from discovering new biomarkers with sets of panels of proteins that are known to be important for a given pathology or disease, and then translating those into smaller subsets of panels that could be used for differential diagnosis or staging disease or predicting response to therapy, for example. And then also being able to run single biomarker assays for clinical use down in the future, like p-tau217 or NFL, for example, in the neurodegeneration field. And you can do all of this with the Argo system, with the same chemistry, and with and go through that whole process seamlessly without having to change the technology. And this is truly unique for the Alamar technology. No other technology can offer this flexibility. Now what I'm going to do is talk you through the NuLISA technology. The NuLISA technology is based on a technology that's been very popular for the last 20 years called proximity ligation assay, PLA. But NuLISA has been advanced and evolved to enable much, much higher sensitivity. And the way we've done this is basically by suppressing background to improve the signal to noise ratio. What you can see in this graphical illustration is the protein that we are trying to measure, the antigen that we're trying to measure in yellow. And then we develop or source two monoclonal antibodies that bind to different epitopes of the protein that you're trying to measure. And both those antibodies have to bind to the protein to generate a signal and an immune complex. So there is an innate high specificity built in to the Neulisa chemistry. We conjugate DNA sequences to each antibody that have a known DNA sequence, which also enables very high specificity, as when you read off that sequence, you know that the right antibodies have bound together to the protein you're trying to measure. What's unique about Neulisa is that we have a poly-A-tailed oligo on one of the monoclonal antibodies and a biotinylated oligo for sequential capture on the second monoclonal antibody. And what this enables us to do is capture this immune complex on magnetic beads and wash away sequentially all of the background before we generate the signal. And so this very simple mechanism of capturing and washing the immune complex is what enables Neulisa to achieve thousands of fold higher sensitivity than a traditional proximity ligation assay. All steps of this assay are performed on the Argo instrument. Step 1 is an incubation of the antibodies for the proteins you're trying to measure into the sample of interest. Let's assume it's a blood sample. And within an hour, these monoclonal antibodies bind to the protein that you're trying to measure. And you can see it forms this immune complex with the oligo or DNA sequences in close proximity or close by one another, but not joined together. We then add oligo DT beads into the well, and this enables that poly-A tail on the first antibody to bind to the beads and capture the immune complex. We then wash to remove any proteins, any matrix, or any unbound antibodies. Then stage 4, we release with a salt wash, which is very mild. We release this immune complex back into solution. And then we introduce streptavidin beads. And these enable us to use the biotin on that second antibody to capture the immune complex for a second time and perform a second wash step. And by doing this, we have a very strong bond between the streptavidin and the biotin, and we can then take this purified immune complex, ligate the ends of the oligos sequences, DNA sequences together, and then amplify them and measure them. And for small numbers of proteins, let's say 1 to 15 in the future, we can use the qPCR readout on the Argo to do the measurement and achieve absolute quantification versus a calibration curve. And then for the broad panels, we can introduce a sample barcode, pool all the samples from 86 samples on a plate into a single library, and then measure them using an Illumina sequencer or an Element sequencer. And we can create a calibration curve to get absolute measurements for hundreds of proteins at the same time. And so this assay background removal step drastically improves signal to noise ratio, and hence provides the highest sensitivity of any proteomics platform available today. This data from our Nature Communications paper shows two experiments that we performed. The first one, using the same antibody pairs, compared IL-4, interleukin-4, a low abundant cytokine, and shows the limit of detection in blue that we were able to achieve with Neulisa, which was 29 attomoles. Using the same antibodies, we also generated data with a traditional proximity ligation assay. And you can see the limit of detection of 389 femtomoles. So this is a 10,000-fold improvement in sensitivity over traditional proximity ligation assay. On the right-hand side, we're comparing an assay we developed for HIV P24 protein with Neulisa and with Quanteric's Simoa platform, which was previously the highest sensitivity platform available. And again, using the same antibodies, we measured the Neulisa assay with the Simoa assay, and you can see that we have a 10 times lower limit of detection and a broader dynamic range, and all using 5 times less sample input. So this proved that we can achieve exceptionally high sensitivity with the Neulisa platform. We have now gone on to develop hundreds of assays with very high levels of rigor and sensitivity. Here you can see 6 important cytokines that are traditionally really hard to measure with traditional technologies. For example, IL-13, a type 2 inflammatory biomarker, is typically below the limit of detection in healthy individuals when measured with Luminex or with Olink or even with mesoscale discovery. But because of the incredible sensitivity of Neulisa, we are able to detect IL-13 very robustly in healthy individuals with really high precision and sensitivity. And we've shown that we can do this now in actually hundreds of different cytokines, chemokines, and neurological markers. When we develop new assays for Neulisa, we go through an incredible amount of validation to make sure that they're both the best in class for sensitivity. Usually we compare it to Simoa, for example, and also the best in class for specificity and performance and linearity. So this example here is for IL-1 beta, another important biomarker that measures the inflammasome. And here you can see we're a 7 times or 7-fold lower limit of detection with Neulisa versus the Quanterix Simoa platform. And also we have a very broad dynamic range of about 6 logs of measurable signal and incredible results when you look at parallelism across a 16-time dilution experiment. And also when you spike in a recombinant protein for IL-1B, you can see that we recover all that protein with very high levels of recovery. And these are all signs that these are not only sensitive, but also reproducible and highly precise assays that are performing at a better level than typical immunoassays. And what's most exciting about Neulisa is not that you can do this with a single biomarker like IL-1 beta, which is still a great step forward for the technology, but now you can also achieve this same sensitivity in a multiplex readout. So here you can see 5 cytokines, including IL-4, IL-6, IL-8, IL-1 beta, and TNF-alpha. And at the top, they're measured with our qPCR singleplex assay. And these are whole blood samples stimulated with LPS with 3 healthy donor samples. And so at the top, you can see the results we got with singleplex. And at the bottom, you can see the results achieved with the multiplex sequencing readout. So all 5 proteins measured at the same time. And so here we're demonstrating the same levels of sensitivity and the same levels of precision and accuracy, whether it's singleplex or lowplex, singleplex or multiplex readout. So this enables Alamar to develop very large panels. And this is our flagship inflammation panel 250. And the 250 represents the number of biomarkers that you can measure in a single experiment with 10 microliters of sample input. And what you can see from this list of biomarkers here is that we're able to measure very low abundant cytokines and chemokines and growth factors that most technologies cannot measure, like different isoforms of interferon, different isoforms of IL-17, markers like IL-33, IL-5, and many very interesting markers related to oncology and inflammation, like PDL1, PDL2, etc. And you can measure all of these biomarkers from a single 10 microliter sample with exceptionally high reproducibility. And what's also unique about this panel is not only can you measure these very low abundant proteins, but also you can measure high abundant proteins at the same time, like CRP. So it enables us to build basically the most comprehensive immune monitoring panel that's available today. And you can measure all 250 of these biomarkers from healthy plasma samples. If you compare this to a platform like Olink with the same biomarkers in some of their larger panels, their detectability is typically 60 to 70% that can be measured in healthy plasma samples. And with NULYSA, we're able to achieve almost 100% detectability of these markers. And this panel was showcased in our paper that I mentioned earlier in Nature Communications. And in this paper, we performed a comparative study with Olink and with mesoscale discovery. And we ran the same set of samples, 151 samples across 2 plates, 79 healthy and 72 from various diseases like cancer, inflammatory disease, kidney disease, metabolic disease, and neurological disease. And we compared the NULYSA platform at the time had 204 targets with the Olink Explore platform with 368 targets. And what you can see here is actually a comparison of the detectability of the NULYSA platform versus Mesoscale and Olink. And many of these assays are measured well by the 3 platforms, but you can see at the bottom a subset of very important markers where detectability for Olink and mesoscale discovery starts to become very poor. And that's where the correlation starts to drop. So typically when you compare Olink to NULYSA, the correlation is very good for those medium to high abundant proteins, but those that are very low abundant can only be measured with NULYSA. On the right-hand side, you can see that we're able to achieve exceptionally high reproducibility, both intra and interplate CVs of less than 12%. And why is this important? Because the biology of these low abundant proteins is often driving disease. And so what you can see here is, again, data from the publication. But here you can see a number of important cytokines and chemokines that are differentially expressed in both the Olink and the NULYSA platform when you compare patients with inflammation disease and healthy donors. But there is a subset on the left-hand side, and you can see them in this lasso plot down here, that there are a subset of proteins that were only seen with NULYSA, significant changes in IL-4, IL-33, IL-5, OSM, that were missed by the Olink platform, simply because it wasn't sensitive enough to measure them in the healthy controls. And we've seen this now many, many times in multiple studies, including this study that was done by Holden Maker from Stanford University and published in the Journal of Immunology. And it clearly shows a 3-way comparison in a COVID study called IMPACT study by the NIH And in this study, they looked at 2,000 patients and compared the expression levels of different cytokines to see which ones were driving disease in COVID patients with different levels of severity. And the bottom line was that the Alamar platform performed with much, much higher sensitivity and precision. 97% of the targets in the panel were detectable versus 79 and 82 for the other two platforms. And Stanford has subsequently become one of our early customers and is now running all of their immune monitoring with the Alamar Nebulizer platform. So now in the last 15 minutes or so, I'm going to change to talk about neurodegenerative disease. Because whilst inflammation is important across many diseases, in neurodegenerative disease, there are specific biomarkers that you want to be able to measure with exquisite sensitivity in blood samples. One such marker is neurofilament light. I'm sure you're very familiar with this key neurodegenerative biomarker, important in ALS, important in multiple sclerosis, and also in Alzheimer's disease. Well, we've demonstrated a best-in-class performance of our Nebulizer assay, which outperforms the current best-in-class Simoa platform from Quanterix, with almost 4 times the better sensitivity, with 5 times less sample input. And we have run many studies now with our NFL singleplex assay, demonstrating very good correlation with Quanterix and the ability to measure NFL in normal Alzheimer's disease and commercial CSF samples. As I'm sure you're aware, phospho-tau 217 is currently the most important biomarker for diagnosing Alzheimer's disease. We have partnered with ALZpath to source antibodies and developed the high sensitivity p-tau 217 assay. And here you can see that we can achieve 6 femtogram per mil levels in limit of detection and 12 femtogram per mil levels of limit of quantification for p-tau 217 in blood samples. So we've really developed an exceptional sensitive phospho-tau 217 assay. But again, singleplex is great, but what you really want to be able to do for neurological diseases is look at the whole pathology of neurological disease. Is amyloid driving the disease? Is tau pathology driving the disease? Is there co-pathology that might be driven by synuclein or synaptic biomarkers? Or is this a vascular version of dementia that has a different outcome? Or are there other neurodegenerative or inflammation markers that can differentiate between different forms of CNS disease? And so now, for the first time, made possible by Alamar's sensitivity and multiplexing, we've developed this CNS disease panel with 120 biomarkers, including all of the key phosphorylated tau biomarkers, both peripheral phosphorylated tau as well as brain-derived phosphorylated tau isoforms. And I will try and explain this in a moment. So you can measure all of these different forms of phosphorylated tau alongside amyloid beta 38, 40, 42, NFL, GFAP, and also all of these additional markers like TDP43 and alpha-synuclein. This panel was actually not designed by Alamar. It was designed by Henrik Zetterberg and by a number of pharmaceutical companies who asked us if we could build a panel to incorporate all of these biomarkers from a single blood sample with high detectability. We launched this panel just over 1 year ago, and it's already been run on over 100,000 patient samples. It really gives you all of the key hallmarks of neurodegenerative disease in a single blood sample. Oops. Now I'm going to just tell you a story about a very new biomarker that we've just developed, which actually allows you to differentiate between brain-derived phosphorylated tau and phosphorylated tau that may originate from other areas of the central nervous system outside of the brain. So tau protein can be distributed in CSF, where 100% of all the tau comes from the brain. And an example of this would be in Alzheimer's disease, for example. But in blood, 20% of tau that you find is coming from the brain, but 80% of the tau is coming from somewhere else in the peripheral nervous system. And an example of this is something like ALS or amyotrophic lateral sclerosis. And so now you have the ability to differentiate between brain-derived tau and tau that's derived from elsewhere in the central nervous system. And this is really important because it enables much higher precision when measuring tau 217, for example, in plasma, compared to traditional assays which measure what we call N-terminal p-tau 217. So you can see here the AUC, the area under the curve, for plasma-based brain-derived p-tau in patients with Alzheimer's disease and amyloid pathology is almost 0.99, which is exceptionally high and equivalent to what you would see in CSF. And this is a busy slide, but basically it shows that you can now differentiate between ALS and Alzheimer's disease by measuring brain-derived tau versus N-terminal tau, peripheral tau. And you'll only see the N-terminal peripheral tau upregulated in ALS patients, for example, and not in Alzheimer's disease patients. So now many leading labs in the world have adopted Neulisa for neurodegenerative disease research. You may know Henrik Zetterberg from University of Gothenburg, Nicholas Ashton, who's now at Banner Health in the USA, and Thomas Karikari, who's at the University of Pittsburgh, and Carlos Cruchaga and Laura Ibanez from Washington University. They've all published papers, and you can click on these links and read those papers that have demonstrated that Neulisa is the best-in-class technology for measuring blood-based neurodegenerative biomarkers. And they've compared our assay to best-in-class clinical-grade singleplex assays using our CNS panel versus singleplex, Quanterix or MSD or other assays. And what they've seen is that the Neulisa assay is one of the best assays available for measuring Alzheimer's disease and amyloid pathology. And in this paper, they compared all of these different p-tau assays together in the same samples from a memory clinic from London. And they saw that Neulisa outperformed Quanterix, even though we have the same antibody from ALZpath, and we're measuring our p-tau 217 in the context of 120 other biomarkers. So we have the ability to measure p-tau 217 with higher accuracy than singleplex, Simoa, or MSD assays. And when you look at a larger study like the TRIAD study from Canada, where they look at 1,000 patients from a memory clinic with really deep imaging and CSF-based biomarkers, what they found was that in terms of predicting amyloid PET positivity, the Alamar technology with our p-tau 217 assay was the best performing assay, and also among the best performing assay for predicting tau PET positivity. So this has proven that our panel performs as well as clinical grade p-tau 217 assays. And what the panel enables, which is truly unique, is the ability to look at biomarker signatures that can differentiate different forms or different stages of neurodegenerative disease. For example, here you can see a specific biomarker pattern in patients that have mild cognitive impairment, and then a very different pattern of biomarkers, including all the phosphorylated tau's, GFAP, NFL, that are now upregulated in patients with early tau deposition in the brain. So now, for the first time, you can truly differentiate between patients with different severity or different progression of Alzheimer's disease. Also within the panel, you can look at ApoE4 status, genotyping status at the protein level. So you can now look at individuals who are homozygous carriers for ApoE4, heterozygous carriers, or homozygous or ApoE4 non-carriers. And so you get this information as well from the CNS panel, which is very, very exciting because now it means you don't have to also do genomic analysis to know who is a carrier for ApoE4. One last showcase is this paper from Tommy Karikari. And I love this example because it compares Neulisa to all of the current clinical assays, but in a pre-clinical cohort, ostensibly patients that haven't yet developed dementia. And you can actually, in this paper, you can see how the different biomarkers that you can measure with the CNS panel enable you to understand much more deeply the diseases that patients are developing, whether it be inflammatory, vascular, or synaptic markers that are associated with amyloid tau and neurodegenerative imaging. There is also incredible correlation between the Neulisa assays and the Quanterix singleplex assays for NFL, GFAP, and p-tau217, and better performance in terms of amyloid PET positivity compared to the Simoa comparison. So this is a really nice study to show how well Neulisa performs. And the conclusions were that Neulisa demonstrated strong correlation and comparable diagnostic accuracy to Simoa assays. But because we have the broad panel, you can now identify new targets that show cross-sectional and longitudinal association with amyloid, tau, and neurodegeneration. And this is possible now to understand what's driving underlying biomarkers and pathology driving Alzheimer's disease in a preclinical cohort. We've also validated our platform on a number of remote blood collection devices. So if you want to not use blood collected in a traditional way, but you want to collect them onto dried blood spots, you can utilize the Neulisa panel and still achieve exceptionally high levels of detectability. And so we're building more panels. We have a mouse panel, we're building a metabolic disease panel, and we're expanding our CNS and inflammation panels over time. And we also have an ever-growing list of singleplex assays that you can access. This mouse panel, the Murine Panel 120, has 120 key biomarkers related to immunology, neurology, and oncology. And it's by far the broadest and most sensitive platform available for measuring biomarkers in mouse samples. And with this particular panel, you can go down to 5 microliters of sample input for blood and CSF samples. We've also recently launched a custom assay development kit. So now if you have antibodies for a biomarker that you want to develop with ultra high sensitivity, you can do this very easily on the Argo instrument using our custom assay development kit on the Neulisa chemistry. So our product menu is growing. We have multiplex panels, we have singleplex assays, we have the homebrew custom assay development kit, and you can also access our services soon through Philokaria and their own service lab. I'll just leave you with a couple of links to webinars that have been given by our customers showing how important the Neulisa chemistry is going to be, particularly for thinking about Alzheimer's disease and going beyond a straightforward diagnosis, but understanding the underlying co-pathologies driving different neurological diseases. This is a webinar by Nick Ashton. At the time, he was at the University of Gothenburg, which really describes very beautifully how the Neuralizer technology is changing the paradigm for neuroscience. I've already mentioned the ability to do dried blood spots, but Henrik Zetterberg has created a wonderful article talking about the use of this Caputena remote blood collection device and RCNS disease panel for measuring really key biomarkers from dried blood spots. And what you can see here are some of these key biomarkers that are measurable from dried blood spots with the Neuralizer CNS panel. We believe this is going to be the future of biomarker detection for screening and for large-scale population screening for Alzheimer's disease. So to wrap up, I'll just give you a glimpse into the future. We received $10 million from the Gates Ventures Alzheimer's Drug Discovery Foundation. And this is to basically develop and fast-track the development of our diagnostic Argo platform. And this platform will enable you to run all of our RUO assays, but also our future diagnostic and diagnostic assays that we intend to develop. So the first screening panel that we will be launching in the next few months is a 5-plex panel for Alzheimer's disease screening, containing the brain-derived p-tau217, amyloid beta 42, GFAP, NFL, and ApoE4. And this panel will be available on both the Argo HT RUO and DX platforms. And then in the future, we will develop more panels and smaller collections of biomarkers important for different neurological and immune disorders. So my final slide is that Alamar has developed a groundbreaking new technology, which is well positioned to accelerate innovation for early disease detection. It's truly a breakthrough platform that combines unmatched sensitivity for multiplex analysis with full automation that allows you to go from discovery to diagnostics. And we've had some amazing commercial success early on in Alzheimer's disease with leading experts in the field that have demonstrated that this is truly a best-in-class proteomics platform. We have the Argo instrument and the ability to democratize the testing by using remote blood collection devices. And the team that Yuling and his fellow scientists have built has a track record of execution and creating value for our customers and for our shareholders. So we're very excited about the future of the Neuralizer technology, and we'd love to work with all of you to do some fantastic science and utilize the potential of Neuralizer. Thank you very much for listening. I hope you were able to understand my English. I'm happy to take questions.